Right. Welcome everyone to this panel discussion in the Genomic Prediction webinar series. I'm Lawrence Tellier, CEO of Genomic Prediction, and I'll be your moderator for an astonishing subject: CRISPR used to completely remove an entire chromosome in human embryo cells for the first time in history. It's a step of the future, but it's now done, and more is coming. So let me introduce the panelists. Professor Simon Fischel, early IVF pioneer, is president and founder of Care Fertility Group. Professor Fischel has worked 10 years with the Nobel laureate inventor of IVF, Professor Sir Robert Edwards, from 1975 to 1985, and was part of the original team of 4 to open the world's first IVF unit, Bourne Hall, outside Cambridge in 1980. We estimate that he has been responsible for about 10,000 babies over the past 40 years. Professor Fischel is famed for his history at the forefront of every major technological breakthrough in the field of IVF, and is the head of the Genomic Prediction Scientific Advisory Board. And apropos the board, we're today introducing Dr. Serena Chen's addition to Genomic Prediction's Scientific Advisory Board. Dr. Chen is the Director of Reproductive Medicine at IRMS and St. Barnabas Medical Center. One reason why Dr. Chen has been recipient of numerous top doctor accolades by peers and patients may be that Dr. Chen is a very prolific and visible actor in the fertility ethics space, including the ethics of the patient's right to be informed and involved in decision-making. The fit with Genomic Prediction is excellent, and Dr. Chen's appointment to Genomic Prediction Science Board is a very proud moment for us all. Dr. Chen is an old friend of Dr. Jamie Metzl, a leading technology futurist and geopolitical expert, a former director on the National Security Council of the United States, a founder of the global social movement One Shared World, and an appointee to the World Health Organization Expert Advisory Committee on Human Genome Editing. He is the author of 5 books, including the recent bestseller Hacking Darwin, Genetic Engineering and the Future of Humanity. It's fitting that these figures are now familiar with Dr. Dieter Eatley, who is among the world's foremost experts in CRISPR editing of the human embryo. Dr. Eatley is a Maimonides Assistant Professor of Developmental Cell Biology at Columbia University and the architect of the study results, which we'll be discussing today. The Eatley Lab is at Columbia also working on mitochondrial replacement and pluripotent stem cells relevant to the reproductive technology applications at hand. And finally, Dr. Nathan Treff is a founder of Genomic Prediction, our CSO and lab director. He's an associate professor at Rutgers University School of Medicine. He teaches reproductive genetics. Treff has published over 100 reviewed papers in reproductive genetics, is a recipient of 2 General Program, 2 SIRT, 2 SRBT Prize Paper Awards, and serves as a member of the ASRM Research Committee, as Senior Associate Editor of JARG, and as Editor of Fertility and Sterility. So Nathan will be introducing the topic at hand with Dieter. Nathan, the floor is yours.
Great, thanks a lot. And thanks to everyone for joining, particularly Simon coming to us from over the pond. I think that's how you say it. Appreciate it. I know it's late, and everyone else, thanks again. Yeah, so just some background. Dieter and I have known each other quite some time, all the way back to when we were postdoctoral fellows trying to figure things out and focusing on diabetes research. We've been collaborating for about 8 years, and I've been really lucky to be a part of the work that Dieter has been doing. And a lot of the success we've had has been related to obviously his work, but the genetic testing has been an important part of the observations made, and it's a big part of what we're going to talk about today. So thanks again, everybody, and Dieter, I'm going to pass it off to you to get started.
Thank you very much, Nathan. It's a pleasure to talk to all of you who are attending this meeting in this context and on this platform. So I've been working with Nathan for a number of years, and this, I think, is a particularly exciting story that we did together with, of course, a number of important contributors and collaborators, and I will mention them in the end of the presentation. So my lab is interested in how can we improve human health in general? And are there ways to do that by intervening at the earliest stages of human development? I believe the beginning is important, and perhaps if something goes wrong in the beginning, it can of course affect the rest of the entire life of that person. And also it means that this is a very powerful window to potentially prevent disease. And so I think there's a great promise. IVF, the area of IVF provides a window of opportunity where something could be done. But surprisingly, perhaps, currently there's essentially no treatments that would do anything to the embryo that will later on improve human health, other than, I would say, would describe them as rather, maybe not a direct intervention, but embryo selection. That's something that's common. But the embryo itself is essentially on its own. There is no treatment for it. And so are there ways to make therapeutic interventions at that point? And we do, of course, know that the genes that we get in the beginning of life, they are very important in determining our health as adults. So throughout life, millions are affected by heritable single gene disorders, and many more are affected by polygenic disorders. And I think we all of us carry risk variants that predispose us to one or the other disease. We may not know all of them, but we'll, unfortunately sometimes find out in our lives. And while we enjoy great advances in medicine and the management of these diseases, the question really is, will that be enough, right? Or can we do more to maybe prevent some of that? So the advantage, of course, would be that you could prevent diseases before irreversible damage occurs. Sometimes once the problem is here and it's realized, it's already too late to fully reverse it. And then furthermore, you can see in the center of this image, it's just one cell, right? So unlike in the human body that has billions of cells, here you only need to treat one cell. So it's extremely effective, or it should be extremely effective. But of course, it also has much greater risk because whatever happens here will be present later on, including in the germline. So it's permanent. It would, if a mutation is fixed, prevent the propagation through reproduction. So it's a permanent fix. It has thereby the significant potential to significantly advance human health. But of course, it also carries the great weight that if something's introduced that's adverse, then how to get rid of it. We wouldn't have an answer to that. And so we really wanted to know how could a mutation, in this case a mutation that causes blindness, be fixed. We were interested in that more from a basic research standpoint because this form of blindness, you could actually solve this with PGD. It's a recessive mutation, meaning that you need 2 mutant copies to be blind. And in most cases, unless both partners carry that same mutation, the offspring would actually be fine with just one of these mutant alleles. So with PGD would be very effective for this. But in this case, this sperm donor had 2 mutations, so was actually blind. He donated his gametes, and we wanted to determine, can we fix this mutation? And so the question we actually had is whether that paternal mutation could be fixed with the maternal genome that carries the normal copy. So essentially, can the embryo fix itself? And what you can see here is that the sperm is injected together with Cas9. It cuts the chromosome into 2 pieces where the mutation is, and then we look for whether it incorporates that normal red DNA sequence from the maternal allele. So this gene is on chromosome 6. It's about in the middle or near the, what's called the centromere of the chromosome. When you design a guide RNA, what you do is you make it specific for that one sequence. So you can see that the red sequence there, these letters, they have in the beginning where the Cas9 cleavage site is indicated, and then this RS, that's called a SNP. This is the actual mutation. There is a G missing relative to the maternal wild type DNA sequence. The maternal one is GG, while the paternal one misses a nucleotide, and that's what's causing blindness.
Dieter, can I ask a question?
Go ahead. Yeah, please feel free to interrupt.
So if you're injecting the Cas9 with the sperm, then the Cas9, then the two pronuclei form and they join to make the embryonic genome, and then the Cas9 works. It doesn't work on the sperm or the egg separately. It knows to work on the 2PN or just after the 2PN is formed.
Good point. I think I have this in the next slide
Okay.
here. So what you see here is that the Cas9 is injected with the sperm, and then you have the egg, and once it's in, everything is together. So the sperm, we don't inject the Cas9 into the sperm. The sperm is kind of, it has to open its chromatin. The chromatin has to unfold for the Cas9 to gain access. The sperm is kind of a cell. It has a membrane, so the Cas9 doesn't just get in there. Even if we kind of base it in that Cas9, there's also no known DNA repair machinery in there. So this process relies on, the Cas9 makes the break, but then the cell has to fix it, right? So it relies on the endogenous DNA repair mechanisms which are contained in the egg. So what you're saying is everything happens after we inject it in the egg. So
And
here
so
you see,
theoretically, hopefully, it happens at the very beginning of when the sperm and the egg genome are together before cellular division occurs, because otherwise you could have mosaicism.
exactly, that's exactly the point. And I think this is really the smart thing to do here, is to inject it as early as possible. So where the sperm has a single copy of the genome, so you only have to really fix one copy of the DNA, right?
Yeah.
As soon as the cell goes through the first cell cycle, it starts to have two.
Okay.
So here, the Cas9 is injected with the sperm, and there's just a single copy. And so the hope is that it will cleave very quickly, and then the fixing occurs very quickly. We find this is in most cases, this is the case, right? And so here what you see then, we let them develop to the cleavage stage, and then the blastocyst stage, and this is where you can do the genetic testing. And this is where we send nascent our cells. You can see here the blastocyst has, a few cells have taken and put into a tube. With the inner cell mass, we can make an ESL line. So we can kind of do two things with such a blastocyst to carefully characterize its genome. And this is how, these cells or that DNA went to Nathan for the assays that I will just describe. And these are below. And I think this is really where, the conclusions of the study come from and are key, is that you do need that genetic analysis in order to know what's going on with what Cas9 does to that genome. You can, of course, look at just a segment of DNA, right? If you look at just the little piece of DNA, that's where you cut, and that's in these peaks that you see in the lower left. You can see here we have kind of a double peak, which is indicating heterozygosity, which means we do see both chromosomes, right? We see the paternal genome and the maternal one. And these double peaks actually tell us that Cas9 did cleave, and it stitched it back together, but in a different way than it was before. That's called an insertion, an indel, usually either a small insertion or a deletion. So that's what you see with that Sanger profile. And when you look at the entire chromosome 6, you see these blue dots in the what it says paternal contribution. These blue dots mean that it is called heterozygosity, meaning we can detect throughout the chromosome, we can detect polymorphisms that come from the father or the mother, or paternal or maternal origin. And it's called paternal contribution, so it's 0.5, meaning half of the chromosomes are from parental and paternal, and half of the chromosomes are maternal. So this is what it looks like when Cas9 makes an indel. These indels are changes that are usually something completely novel that has not existed in the human genome before. So that's not something that we think could be useful clinically, because how do you know whether what that changed DNA does, right? You don't. So there is, just recently was a report from the national academies, and they state that only variants that we know are very common should be considered for introduction, right? Common in the population, and we know what they do to human health. In this particular new variant that was Cas9 generated here, we wouldn't know that. Right? So this is good enough for a stem cell, but it's not for reproduction, for reproductive purpose.
Right now, there's not a good way to say, okay, we're going to make sure the DNA goes to exactly the right place.
So when the break is introduced, then the cell chooses what to do. It has multiple options, and this is one choice that it sometimes makes, and actually fairly often, in about half of the cases, it stitches it back together in a way that is kind of imprecise and induces small insertions or deletions. And that these are alleles, these are altered DNA sequences that we don't fully understand, and so it certainly wouldn't be a wise idea to have a human being from such a changed allele, right?
Right.
So I think this makes sense. I think this is what most people would agree with, and I mean, there's always a discussion. This discussion is still very young, all right, of how would you do that, but I think this is certainly very prudent not to introduce any novel allele that we don't already know what it does in a.
What happened with the Asian babies that were born? Like, do they somehow got that gene to go into the right spot, or we don't really know?
This is exactly the process that occurred in those babies. Some people call them the CRISPR babies, however they are called. So the two children that were born in 2018, they had this process, a small insertion or deletion that is not existing in the human population, right? So what's the consequence of that? We don't know, and maybe we never will. It's of course possible that there is no consequence, but why would you want to find out, right?
Right.
You want to find out. You want to know. I mean, medicine is all about knowing what the consequences are that you're doing, right? I mean, that's at least my understanding of medicine. You need to know what
weigh the risks and the benefits. Yes, I agree.
The benefits. So what's the benefit? We don't know, and so it's better not to do a new novel version that you don't already have in the human population. And I think that's going to be the consensus, right? I think it also really makes sense because you don't want to introduce anything new. Nature does that sometimes, and sometimes it can be detrimental, right? So we don't want to play with that. But these are certainly very important talking points. So the surprise came when we looked at a few other blastocysts. That's what these structures are called in the upper left. These blastocysts looked, you could say, fairly normal. You couldn't tell morphologically that they were in any way different from the other blastocysts that I just showed you. But we saw that they are wild type. If you look at this profile, here that's called the Sanger profile, where it says EYS wild type, so EYS is the gene. The wild type means this G is present. If the mutant doesn't have that G, right, it's deleted. So here you have an embryo that looks wild type, completely wild type. So you could think of, wow, great, we've corrected that mutation. And there's some people, it seems like that. So this is what we were looking for and we're hoping for. It would of course be tremendous if that's what it would be. But that's why you also have to look at the entire genome. And this is again where that SNP platform that Nathan is running became extremely critical to understand what's happening here. And so I have to explain this a little bit, right? Again, the blue areas, this is where you have both a maternal and a paternal genome. And now you see this drop exactly where that arrow is. The arrow points to where the chromosome is cut, right? So from left to right, that's chromosome 6. And where the arrow is, the big arrowhead, that's where Cas9 did cut. And you can see all the way on that other side, this chromosome misses all paternal contribution, right? It's zero paternal contribution, it's only from maternal. Meaning Cas9 cut off an entire chromosomal arm. And so, you know, you wouldn't know that if you just looked at the site that you target with Cas9, because that site looked wild type and it seemed like we had corrected the mutation. That wasn't the case. Now you go down and look at E, and there's another embryo that's wild type. Again, you could think that you had corrected the mutation, but again, that wasn't the case. And instead, the paternal contribution was gone entirely along the chromosome. You can see that there is a lot of red dots at the bottom where it says zero paternal contribution. There's a few blue dots, and there's also a few green dots in some areas, but we believe that there's no DNA there from paternal contribution, that these are artifacts of genome amplification, essentially. So, but we do know, we can see here that the paternal contribution is extremely low, so that means it's 0, because you can't have 3% of paternal contribution. It's either 50 or 1 or 0. So we do know that the maternal contribution is 100% along the chromosome, and so an entire chromosome was lost. So in the context of correcting mutation, this is very undesirable, right? First of all, you couldn't use such an embryo for reproduction because it's not viable. It cannot live without a chromosome 6. Right? This is a deficiency that will ultimately result in, you know, the loss of that embryo. So this will not develop. You know, if it were to be attempted, which of course we did not intend at all, and this is purely for research purposes, but if it were to be attempted, what would happen is that embryo would probably not implant, or it would demise very early on. But fortunately, we looked at this, right? And so now we do know something, an outcome that we didn't know before, because as you point out, there had been efforts to make children from this, one in Asia, and there had been people in Russia thinking quite seriously to use this method to introduce changes in the genome elsewhere. And, you know, knowing this, I think it will give them some thought and to reconsider. You know, whoever might consider doing this in clinically will look at those data and say, well, wait a minute, if I did this, what would happen, right? The outcomes could actually be quite detrimental. So in the context of a mutation correction, this is not what you want. But I think we learned something tremendous here. First of all, we learned that the human embryo is not very good at repairing such a break, right? At the top is this break has been around for quite a while in this embryo, but it just failed to seal those chromosomes back together and have 2 normal copies of that chromosome 6. And this was really a surprise that the human embryo fails to repair such a break. And the second,
Dieter, quick question on that. Had you used base editing or prime editing, do you think the outcome would have been different or not?
I think this is really a great question. So base editing or prime editing doesn't automatically induce a double strand break in the DNA. It induces a single strand break. And so, but it still relies on the endogenous repair machinery of the embryo to then seal that back, that single stranded break. So we haven't done this, and surprisingly, it also hasn't been looked at in other studies. And you know, this is precisely why basic studies are so important before anything, you know, is attempted clinically, because you first need to have these basic studies to know what the consequences are. How does the human embryo deal with such a break? How does it repair it or not? And only then you know what to do with the tool, right? Right now, this is just a tool, and you can have very important basic research insight, but you know, it's clearly too early too. And you can take the time and think about what could this be useful for, right? But the application is clearly too early at this time. There is a lot that needs to be done. We can get back to that discussion later, which I think is a really important discussion. So, but what we learned here as well is that you could actually remove a chromosome with just Cas9. Another thing that we found is when you looked more closely at the embryo and the entire genome, we also found changes elsewhere. In particular, so chromosome 6, of course, stood out. So this green high bar, these are the changes I just showed you. This means we most commonly saw changes on chromosome 6, either whole chromosome losses or parts of the chromosome were missing. But we also saw that at one site on chromosome 16. And at that site, there is what's called an off-target site of Cas9. So you can see the guide RNA that we designed. It's about a 20 nucleotide sequence long segment, has a closely matching segment on chromosome 16. Yeah, just as two mismatches, right? And they're located a little bit to the right side of that guide RNA, so in that area it doesn't seem to matter as much. And so what happened here is that despite that mismatch, Cas9 also targeted that chromosome. And again, at that location, we did find some chromosomal changes. What you see here, for example, here it cleaves on chromosome 16 out here. And when you look at this, you can see here is only paternal DNA, so it lost the maternal part. You can also see this blue line, this is the copy number, so there's a clear drop at that location in the number of, in the signal intensity, which means there's less DNA. So there's only one copy of paternal. Here there are two copies, maternal and paternal. And in this cell, we found loss of the paternal copy, and it's only maternal, and again, a copy number loss. So this kind of schematically shown here. Cas9 is injected. It cleaves off the chromosomal ends at the next cell's division. Some of it gets stitched back together with a short indel, and the other chromosome misses a segment. And so here, for example, we know this is the original DNA sequence, the Sanger profile. You can see here, this is an indel, here a small deletion. You can see here that it deleted 5 base pairs relative to this. And in this instance here, it deleted 1 base pair relative to this. You can see here, this G here is missing down there. So we do know Cas9 acted on all chromosomal arms. And these are kind of the portfolio of outcomes that you can get, right? You can get 2 different indels here, or you can lose this chromosomal arm or that one. So this really illustrates the consequences of Cas9 activity. And with this we wouldn't have been able to do that without this platform, because and it's really cool because you can look at the entire genome and you can look at heterozygosity. Something that was also really critical is that we did need to know, you know, what's of maternal or paternal origin, right? And so that's some sophisticated bioinformatics, but it's really phenomenal, the insight we were able to gain here. So what it meant is that we had at that side, we had off-target indels, and we also had off-target aneuploidies. So these are 2 additional consequences that Cas9 can have in the human embryo.
And Dieter, when you say off-target, it's like just completely far away from the target mutation.
Exactly.
Okay.
Let me go back 2 slides. So chromosome 16, we targeted the site on chromosome 6, and so the off-target site is on chromosome 16.
I see. Okay.
So these here, this is 1 chromosome. The human embryo also spontaneously acquires some abnormalities. You can see here that these, but they are relatively rare and they're not repeated. For example, we found one that has an abnormality here on chromosome 10 and one that has one on chromosome 1.
But those were not related to the Cas9.
No, these were not. These were spontaneous. So the human embryo does have an issue with double-strand breaks, also spontaneous breaks, not just the ones induced by Cas9. It seems to have difficulties also repairing spontaneous breaks, and that's what you see here. But when you do Cas9, you repeatedly introduce them in the same location. So that's why chromosome 6 here stands out at EYS. This is the on-target side. That's the one we wanted to cleave.
Okay.
But then here in chromosome 16, that's the off-target side. This is the one, we didn't want to cleave, but Cas9 cleaved it anyway, because you have this, close to perfect match, two nucleotides different. Right?
So Dieter, could you imagine now or in the future doing a different type of design for the guide RNAs to at least prevent the problems on chromosome 16?
Yeah. I mean, not all guide RNAs will do that. You can, another guide RNA targeted to a different site may not have any off-target effects. So here we had one. You could say we had just one. The genome is so big that chances that you will get one are actually quite high. But if you take a 20 nucleotide sequence, usually there's just one perfect match in the genome.
Right.
It
So,
depends how many imperfect matches you have. And there's not, there's entire groups that study off-target effects in somatic cells, so improving it.
Yeah, I mean, it seems like with each one of these each one of these problems, if you take it on its own and say, all right, well, this process, there was the problem of perhaps the double-stranded break was too aggressive. Maybe there's another problem that the guide RNA, we could have had some different formulation. I mean, here it was almost a perfect match. So can we imagine a guide RNA that would have had a higher standard and only accepted a perfect match?
No, absolutely. I mean, there's a number of things that can be done. This, what I'm showing you is just the state of the art today, right?
Yeah.
And this is one study. So,
yeah.
I think there will be more studies in a body of work. And there are ways to improve guide RNAs. We could have moved it perhaps to a different position, and then maybe we wouldn't have any off-target effects. Or some people say that if you shorten the guide RNA, instead of making it 20 long, you make it 18 long, so then the mismatches that are there become more important and are more detrimental to its activity. So there's a number of things that can be done, but unless you look and you do the experiment, you don't know, right? And I think that's really the value of doing such studies. It's not possible to fix mutations in the germline without doing the research to know what the consequences are. Now, we learned a lot of things from animals and from animal models and from cell lines, but you can't infer based on those alone. You have to know what the human embryo does with the break. And
So
here we had
is that in the animal data, we don't see off-target aneuploidy? Like, is this uniquely human?
It's a really good question. So we didn't actually include studies on mice in this paper, but from reading the literature, that people don't see aneuploidies induced in mice, certainly not at that frequency. This is an extremely common event. It seems to be the induction of aneuploidies at that rate is uniquely human. Now, again, I think there need to be more studies on that and maybe more comparative studies, but there's no report in mice that shows something similar, except there is one where they used a multiple guide RNAs, cutting many sites in the mouse genome, and then they also saw some chromosome losses. But it took a lot more cutting. And here it's just a single cut that very efficiently results in the loss of a chromosome. And I think that was, was completely unexpected. That's why also I believe.
No, it feels like the human aneuploidy is so incredibly frequent. Is that something we see in other animals, like such a high rate of aneuploidy in the embryonic stage? Because I feel like even our donors, 50%, the 30 to 50% aneuploidy rate in a 25-year-old donor, it just seems like humans are just super inefficient.
Oh yeah, I mean, you experience that quite often. I mean, this is really, I think, one of the main obstacles of reproduction that you bring up here is, right, the early development is so inefficient. And I think this is part of the reason. How much it explains it, we don't know. But if you lose part of a chromosome or an entire chromosome, it can't be very good. It's bad for development. And it happens very commonly. And I think we humans really stand out here is that we often lose the chromosome early development. And I think this does explain, this could be the process, this could be the principal process that leads to chromosomal loss during early cleavage divisions. Again,
what are we trying to, obviously down the line, downstream, we're trying to achieve more than anything, reproducibility and safety. An understanding that we can do something and it works, and what works is hopefully safe. And this raises so many questions. But before I come on to one I want to ask about the animal, I want to just ask a specific question that the embryologist might be interested in, and that is, if you do this at the point of syngamy, is it actually then a different mitotic process than if you did it at the next stage, at 2 cells, for example? Are you going to get the same result? Because that then asks a different question. Understood.
Again, great question. So we did it both at syngamy at the time of ICSI, and we did it also at the 2-cell stage, and the result was the same. So
right. So if the result is the same, then are you getting the same result in the cells that you get at the blastocyst, or do cells themselves differ? Are you getting a kind of a mosaic outcome?
Yeah, you're totally right. You do get a mosaic outcome if you inject later. Again, that relates to the fact that you have start with several copies that you want to correct, right? So as soon as you start with multiple copies, you have to have an extremely efficient process and get each of them right. Now, if you do it when there's just one copy, you only have to get one of them right? So later on, inevitable mosaicism, essentially inevitable. And early on, I think there's a way to avoid mosaicism. In fact, some of these blastocysts were non-mosaic for small indels or for the loss of an entire chromosome. So you do get non-mosaic events. So in one way, that's good. We do know that now. Cas9 acts extremely early when you inject it. It acts in the first cell cycle. It acts often before there are 2 copies, even before the inner replication. But the challenge is really that it has multiple options of resolution, right? It can make an indel. It can not be repaired at all and result in a chromosome loss. It can create various types of indels. And so the challenge is 2 ways. Well, avoiding mosaicism, avoiding off-target effects like this one here, and then also being efficient enough early on with a specific repair pathway, right? So these things would need to be figured out. Now, that's not going to be done in a day or tomorrow. That requires a good amount of work.
So, Dieter,
Sorry, just to finish off the point that Serena made as well, and I think that is crucial to understand. If you did exactly the same experiment on another species, are we seeing the same result? Or are we looking at a human problem every time? So in other words, we're always going to have to replicate everything in the human and know that is reproducible for the human. Or do we have species differences in outcome when you're injecting at the zygote stage?
I think we will have to do it in human if we want to use it in the clinic, without question. I think there will, model organisms can supplement that, but I think there's no way to fully know what's going to happen from an animal model. If you, I do know that in mice we do know that chromosomal aneuploidies occur much less often. And so far, there are no reports of a single break inducing chromosome loss in mouse embryos. It's possible that some other model organisms, like maybe cows, I've heard, have quite high chromosomal aneuploidies in early preimplantation development. Maybe those would be suitable, right? So hopefully somebody is going to take that on and maybe make a study in that organism. We'll certainly learn a lot of exciting things.
Yeah, so it takes the ethical point. It just takes that ethical point, doesn't it? That you can't avoid the basic research on human embryos.
I don't think you can avoid. I think it's an ethical point, but I think the ethic is strong in favor of it, because this here you're trying to help an embryo. You're trying to fix a mutation that could cause severe disease. Now, we know so little about the human embryo, very, very little. We know much more about worms and flies and mice and their embryos. Flies we won't be able to use to treat human reproductive disorders in any, it doesn't come close. So there's certainly a lot to learn from these model organisms as well, but they can't, they're not sufficient. And it's kind of sad that we know so little about the human embryo and that we have this as a surprise. Because we know so little and about the mechanisms, there's also not anything we can do to help, right? The human embryo either develops or it doesn't. And if it doesn't, that's it, right? If it has a mutation, it has a mutation. That's it. What can you do, right? Nothing. So I think it's really sad that this is not any different. I think the reasons are very compelling to study this. And in fact, and I think many would agree. Of course, I do understand that many people are cautious about that and are hesitant to because of embryo is something special, right?
So because we're here hosted by Genomic Prediction, let me ask you, Dieter, the sequencing question. I mean, you sent the cells to Nathan for analytics. Was there anything different? I mean, I know the Genomic Prediction has their algorithm for predicting what happens later in life. In the sequencing that allowed you to look differently or better at these cells than had you sequenced them or we sequenced them in some other way?
Yeah, I think you look at the entire genome, right? We of course, the essays you see on the right here, these peaks, and I really like them. They're kind of pretty, right? They show the location of where you introduce the breaks, so very localized. You look in that neighborhood of the Cas9 activity, but with those SNP arrays, you can look at the entire genome, and only in combination do you find out what's really going on. So it's extremely powerful. It's been really a joy to look at those data. And I think when it comes to reproductive use, I think probably Nathan will add some more to that, and how this can be very powerful for that. But in this case, it's just a really powerful research tool. Right? So let me, and I think there were some very strong responses to this. The work got covered in various news outlets, so this was exciting to see. It was mostly covered in a way is that, well, look at this, we can't fix mutations yet. So it was mostly looked at it from the perspective of being a disadvantage and being a detrimental outcome. In the context of the mutation correction, it is. But there's a little bit of bright spot on this as well, because essentially what we do know is that sometimes the human embryo does have a chromosome too much, such as chromosome 13 or chromosome 18. And so what we are thinking about is, well, maybe you could correct that, right? Maybe you could remove a chromosome 13 or 18 because, pregnancy failure, miscarriage, right? If you can treat an embryo that has this additional chromosome, that would be fantastic. We have here a number of scientific conclusions, and we may or may not go into each of these. But one thing that's also intriguing is that we can, the human embryo really has an issue in fixing those breaks. So what does this mean for our biology? What does this mean for us as a species? Why is that the case, right? We just learned something really fundamental. What do we learn about us, right? In one way, this affects all of us, either in one way or another. It really does. So I think it's important to understand. It's wonderful we found a way to discover this.
I always wondered about the size of the Y chromosome. That, supposedly the Y chromosome in the ancient, I don't know if this is fake news or not, the Y chromosome in the ancient Egyptians was had a lot more DNA, was a more substantial chromosome than the modern Y chromosome. And, maybe somehow this is the short Y chromosome is related to the control of, the TV remote control. I don't really know, but how did that come about? Is that somehow related to some of what we're seeing here, like the instability, like that we could see, a big change in the genome over a few thousand years, so which is I guess revolutionarily a relatively short time span.
I don't really know about the chromosome of the Egyptians, but
So maybe that's
interesting.
I don't want to misrepresent anything. Simon, do you know?
Yeah, it's obviously an off-target effect of the ancient aliens that were messing around with humans.
So I have, in addition to the ancient Egyptians, I have a question related to what Simon said about, and both of you, on the availability and the desirability of doing this kind of research on human cells. Because if we were having this exact same kind of conversation, but we were talking about other technologies, non-human technologies, what we'd say, all right, there's a problem, let's break it down, and we have two primary modes of figuring this out. One is the theoretical mode, using our principles and logic, and the other is tinkering and experimentation. So if we were building some kind of new transistor, we'd do our analysis, and then we'd have a bunch of different samples. If we were talking about a wheat genome or whatever, we would do it the same way. So I don't know, there's not an answer to this question, but it seems that to make progress, we're going to have to figure out these very complex systems theoretically and conceptually and based on some preliminary rules. But there's going to have to be, it seems, a lot of tinkering like you're doing here to kind of figure out what works, what doesn't work, and we'll have some answers that we wouldn't have predicted, but that doesn't mean they're inexplicable. So how do we think about kind of the principle-based research and tinkering when human cells are involved?
Well, I guess I really feel like the patient, the public, and Jamie, I know you talk about this all the time, the public and the patient voice have to weigh in. This cannot be an isolated ivory tower scientific community discussion. And Simon, of course, like in the HFEA, when they figured out mitochondrial disease treatment using, pronuclear transfer and some of these other, fairly aggressive embryonic cellular, genetic manipulations, there was supposedly tremendous public support for proceeding with that research in humans. And that's part of why the HFEA decided to move forward with that studies. Of course, we're held back here in the United States by moratoriums because of various, legislation that's come through Congress, but the HFEA had a process where they actually somehow polled the public and concluded that there was tremendous public support for really, this is embryonic research, germline genetic modification, a type of germline genetic modification.
Yeah, I think it's a really good point to bring up the UK. I think they've done a tremendous job at this. Really good example, perhaps. But I would like to emphasize that basic research, of course, needs to be very advanced before you can start to do clinical. It's almost not a clinical research anymore. It really has to be in place, right? You have to, because the stakes are so high when a human child is made. So I think the basic research often also uncovers two things. It covers opportunities and limitations. So, for example, here, this clearly is a limitation, right, of the technology, which we didn't know before. So we do need to know that because it helps to avoid both premature and inappropriate use in the clinic as well, right? Now you can regulate a lot of things, but something that's very powerful is knowledge of facts, and somebody who knows that no matter where they are or has access to this information will pause and say, well, this is probably not quite ready for use because I don't know what the outcome of a double strand break is. It could be this or it could be that, or they showed it could be this, right? It could lose a chromosome. So no matter, there's certainly people who wouldn't be in support of this type of research on human cells or human embryos, and I do understand that. But I think no matter whether somebody is in favor of this type of modification in the human germline, I think one can be in support of the research because it does really discourage and help to channel it to only what will be appropriate. Right? Here, clearly, we pointed out a limitation. Our purpose was not to make a human child here. We pointed out a limitation that's very important in informing what will be done, and equally important what will not be done.
So Dieter, connected to that, someone somewhere in China has in their possession the whole genome sequence of the 3 Chinese quote unquote CRISPR babies. What's your best guess if you were to gain access to those files? What's your best guess based on this research of what you'd see?
Well, I mean, development selects for normal chromosomal numbers, right? So they will have the whole genome. Likely, if we had a look at the primary data on embryos, likely some of these embryos would be missing chromosomal arms or entire chromosomes. So we don't have access to the entire study or the entire information. I don't know whether they looked for off-target effects in those children, but I do know, what we do know is that at the target site, they have a deletion that we don't know what the consequence is. And I don't know whether we're going to find out, because you might have to follow lifelong, whether they're willing to do that or not, and whether this is even appropriate or not. I don't know that. So I think the way, these things need to be done in a, you cannot learn enough from an effort that's done in the shadows. It has to be efforts that's endorsed at the highest levels of governance and regulation, or it can't be an effort at all because you wouldn't be able to follow it, right? You wouldn't be able to learn what you do need to learn and what you do need to show. So I think this is unfortunate event.
I did have a question, Dieter, that I think that brings up where you're looking at the trophoblast. Did you ever do any studies where you looked at every single cell in the embryo to see what the distribution of these deletions or additions were? And then the second question I have is anybody on the panel aware of what is happening with the first CRISPR trial in the United States looking at T-cell lymphomas? Like when they're trying to modify the genetics of these T-cell lymphoma cells, are they seeing these kinds of consequences as well, or are they even looking for them?
So I think, again, this is probably quite specific to the embryo, that you have these losses in the embryo, but not in somatic cells. So when we did this same cut in ES cells, and we didn't see the same outcomes. So even just a few developmental steps later, embryonic stem cells, we don't see this anymore. It's really something that happens in the earliest stages of human development. Also concordant with the time when you see these spontaneous abnormalities in karyotype.
So could I just go back and just on this point of regulation, the ISSCR, the International Society for Stem Cell Research, is having to review these guidelines. Because of the work that you and Genomic, really important work, do you think at the moment we have guidelines enough for even the researchers to provide the right information for everybody to understand whether or not there is an anomaly or a normal situation? Because it's in a sense, it's not just what might be a gross outcome for a child and for clinicians to understand this. It's whether the scientists are doing the right analysis, and are the guidelines that we're about or are being revised going to be strong enough to guide the scientists on what to look for?
I really like this point. This is great. The reason is, you do need peers to look at what you do, right? Of course, this is why we publish. When we submit a paper like this one, people criticize what we do and say you should do this, you should do that. But how science works is often how peer review in NIH panels, in grant communities, a competition also between colleagues and peers. And so I would say, while I believe in our study and I think it's well conducted, but I believe that the standards of research in this field suffer from this, that we don't have the NIH peer review on this type of work that could really guide and direct things. And we don't have FDA oversight that would help with any sort of clinical translation. So the absence of all these things actually ultimately the promise is clear, and somebody's gonna step in and say, I want to go ahead. And we know that already happened in China. So somebody felt it's time to move forward. And so we would, I think research and guidance like that needs to be in place. There needs to be a path to appropriate use to discourage the inappropriate use.
Yeah, so my final point on this, I don't want to hog this, but it seems to me that actually the only place to really try to control this at the ultimate level is the journal or the editors of journals to say that they won't accept information unless all that information is available. It's almost the point that Nathan's been making for many years about having the DNA available for others to analyze, because it's quite easy for us all to be a little bit bamboozled and to be romanticized by data which some scientists are saying prove that this editing that they've done gives us a normal feature. But you've actually shown very clearly that that's only in one profile it might look normal. And so therefore there has to be some access to independent analysis of the whole data, as Jamie was saying. But where is that going to, I can't see something like the HFEA, for example, they look at publications to provide the evidence. But it's the publication, if it's being relied on, is it correct? How do we know that? But that's a conundrum for me.
Yeah, I think we'll only learn from a body of research. I mean, there's really good groups. There's few, but there's good groups everywhere in the world. In China, there are people who are doing great work on this. In the UK, there are great group doing work on this. But there's not many. And in the United States, I think there's few. There needs to be a body of work, right? Clearly, the most studies have come out of China. They have also been very early in this type of work. I think there was this one unfortunate event, which I don't expect to be repeated, but I think there needs to be a body of work. And you really pointed out, we do need, you only know what's right if you have it from multiple groups, if you have it repeated, if it becomes reproducible in different hands. I think that's just the process that it will take. And so this will take time.
Well, this is very exciting and fascinating stuff. And what are you planning next, Dieter? Like, what's the next step after this? Are you going to try these single single cleavage tools instead of CRISPR, maybe that would be less destabilizing.
So what we'd like to do, we would like to get our hand on this problem. Right, so this is looks at maternal age, and you can see here that like after 35, the number of eggs or embryos that are euploid, meaning they have a normal number of chromosomes, is decreasing. So the curve that goes up means there are more uneuploid embryos, right? At over age 40, you know, it's more than 20 or 30%, rises up to 50 or more percent. So that is a major challenge for reproduction, right? If somebody wants to have a child at that time, there are fewer eggs and many of them are aneuploid. So the question is, can we fix some of those? That's the problem we would like to get our hands on. And what it would do is we now know that we can eliminate a chromosome with Cas9. So how could we remove like a trisomy 13 that would result in the loss of that embryo? So again, you know, that would involve genome analysis, Cas9 injection, and then biopsy. Again, this would be entirely, you know, in vitro with stem cell derivation. So then you can show that you have succeeded. You can look at the entire genome. Again, entirely a research project, basic research. So I think that would be quite exciting if it's possible. We don't know yet, but I think we'll find out.
So you're thinking that you would do the polar body PGT so you could detect M1 aneuploidy and then inject the Cas9 at a very early stage so that,
Yeah, right.
okay.
You would have to inject at you would have to inject after fertilization because you need the second polar body, but you would have to inject pretty soon. So
Simon, you must love to see the word polar body in future research.
Yes, well, I'm a polar body fan myself. But of course, the thing that also worries me about the older eggs is the mitochondria. And yes, we might be able to one day get a consideration for the gross aneuploidies. But, and we will make eggs become embryos that are more viable. But to create them more efficient, a higher percentage of good eggs, if you like, is whether or not we need to take into account the mitochondria. And if we do, then we may have an issue with really older eggs. In other words, you take a 45-year-old that's got a couple of severe aneuploids, you correct them. Still, what is the potential of that egg? But that's another story. I've thrown something into the bicycle stroke. Unintentionally. But yeah, I think you know it's a real challenge for us, human aneuploidy.
Well, it's one of our greatest barriers now. We need you guys to figure out how to make more eggs and make them normal. So that's what we're looking.
So Serena, I think the other thing that's this is why I love the work of genomic prediction. Understanding more about SNPs and their effect on quality, chances of miscarriage, other reasons of failure. We have been quite rightly so linked to aneuploidy since those very early papers by Angel back in the beginning of the 80s, showing with absolute shock and horror that the first human embryos they were looking at were aneuploid at a very high rate, which we didn't know, we didn't understand. That's been a huge focus for us because, you know, big cause of miscarriage, etc. But even then, you've still got a high incidence of failure. And I think over time, the work that genomic prediction and others will be doing that actually go deeper into the chromosome is going to be so important for us.
Yeah, for things like why do, you know, why would somebody who's only 33 years old without any risk factors have diminished ovarian reserve, right? What produces the egg? Some people have a ton of eggs, some people have very few, and we know so little about that. And endometriosis, polycystic ovarian syndrome, all of these things are big black boxes for us right now. And I think you're right. I think a lot of the key to a lot of this is going to be genetics. So
yeah, and the other thing that we used to call it the occult gene, but for years, and I don't know if research has changed since I looked at it, but if you had sister-to-sister donation, because one of the sisters had poor quality eggs, your overall statistical outcome was lower than if you had another donor.
right.
And it wasn't aneuploidy.
Yeah, and yet the pregnancy-induced hypertension rate is higher with anonymous donors than with sister-sister donors. So that's another interesting thing. So there's a lot of cool stuff for you guys to figure out. But before we go, we have to take a nice picture together. Everybody has to smile. Say cheese.
Jeez.
Good.
So are we done, or are you going to present something, Nathan, or are we done?
No, I think we're done. Thanks again, everyone. Lauren, did you want to close?
Sure. This has been a real pleasure. Thank you all so much for participating in this first panel with all of you together. It's been a real pleasure.
I want
Thanks,
one
guys.
of Lauren's jackets. I want a jacket like Lauren. I like that.
Yeah, I think we all do. I think we all do.
Yeah.
All right. Great job, Dieter. Great job, everybody. Thank you.
Yeah, don't
Thank you.
Keep up the good research.
Yeah.
amazing work.
Excellent. Yeah.
All righty. Bye.
A privilege.
Bye.
Look forward to the next session. Bye.
Bye. Bye. Bye.
Bye.
Bye.